So up until this point, we've been looking at small molecules. Now we're going to look at a group called macromolecules. And macro here means large. So instead of just one small molecule, we're looking at really big molecules. And there are 4 groups of these. And the first one here is the carbohydrates. So take a second and think about what do you eat? What kind of foods are carbohydrates? You probably thought of things like rice or bread or potatoes or starches, sugars. Another source of carbohydrates are fruits and vegetables, right? They have sugar in them, but they also have something called fiber. So fiber is a carbohydrate. Carbohydrates gets a bad rap, but it's really highly processed carbohydrates that tend to be not as healthy for you. So carbohydrates are hydrophilic, which means that they like water. So these are going to dissolve well in water. Like if you put sugar in water, it'll dissolve. And they typically have C, carbon, hydrogen, and oxygen in this ratio of 1 C, 2 hydrogens, and 1 oxygen. A monomer, right? So a single molecule of a carbohydrate is called a mono, 1, and then saccharide. Saccharide means sugar. A dimer, so dimer means 2. So this is going to have 2 carbohydrate molecules. It's called a disaccharide, again. So literally means 2, sugar. And then polymer is going to be called a polysaccharide. So this literally means many sugar. So this is a pretty consistent naming scheme. So your book gives us several examples. So this column over here, these 3, we have monomers, which are glucose, galactose, and fructose. Those are the most common found in our body. Glucose, we're going to see a lot, is C6H12O6. You don't need to memorize that right now, but you will eventually memorize it just by being in this class. The dimers are right here, right? You can see how these all have 1, 2. Sugar molecules joined together by a bond. So we have sucrose, so that's like table sugar. Lactose, so that's the sugar found in milk. And then maltose is another common one. And then our polymers are over here. And we have many polymers. So glycogen is how humans store sugar in your liver and in your muscles. Starch, so these are both examples of starch, amylose and amylopectin. This is how plants store sugar, so things like carrots and potatoes and onions. And then cellulose, right? This is fiber. So you can see how this is really, really well organized. So you can pack a lot together here in fiber. And it's really, really hard for animal digestive systems to break down. And so most of our fiber actually goes undigested. And it's very healthy for us. It helps move your digestive system along. So this is the kind of carbohydrate you're going to find in things like vegetables, celery, the stems and leaves of plants. The second group of macromolecules is called lipids. Lipids are a little weird. They don't have a true monomer, dimer, polymer scheme like the rest of these groups are going to have. Instead, lipids have one characteristic that defines them. They are all hydrophobic, which means that they do not dissolve well in water. So there are 3 types of lipids. The first one are called triglycerides, which are called fats. So if you think about the foods that you eat, what kind of foods are fats? Hopefully you're thinking of things like butter and oil. And then think about if you put oil in with water, it's not going to mix up very well. So this is a fat molecule. It's made up of a glycerol. Here's my big glycerol. And then 3 fatty acids. 1, 2, 3. So if you think about the fats that you eat, we have saturated fats and unsaturated fats. And arguably one of those is better for you than the other. The good fat, quote unquote, I'm doing air quotes, is unsaturated, right? So this is the good fat. Things that you're going to find in plants like avocados. Saturated fats you're going to find in more processed things like butters, nut butters, cheeses, dairy, that kind of thing. So the difference between saturated means to be full of, right? If I saturated you with water, I would probably just dunk you in some water and you'd be saturated. In this case, it means saturated with hydrogens. So this top bright yellow fat here is a saturated fat. You can see how every carbon has all of its hydrogens. So there are all single bonds here. An unsaturated fat means that we have some room here where there's not hydrogens. And instead we end up with double bonds. What happens is that double bond gives the fat this kind of V shape, and they don't stack together very well, and so they don't become very dense. So these unsaturated fats tend to be liquid at room temperature, like an oil, and these saturated fats tend to be solid at room temperature. The next type of fat is called a phospholipid. So a phospholipid consists of two main parts. It's got this hydrophilic head region, which means it likes water, and then this hydrophobic tail region, which means it doesn't like water. We have a special word for something that has both an area that likes water and an area that doesn't like water, and that's called amphipathic. So this allows phospholipids to form cell membranes, which we'll cover more in the next chapter. The third type of lipid is called steroids. So steroids always look like this. They have 1, 2, 3, 4 rings. That fourth ring is always 5 carbons, and then they have these functional groups off of them. So steroids can be things like estrogen and progesterone and cholesterol. You can often see people in sports who take anabolic steroids, and so those would have this structure here, right, as hormones to help them build up muscle to be better. But anabolic steroids have some really, really bad side effects for the reproductive system, as well as some behavioral side effects like extreme anger. The third big group of macromolecules is proteins. So back to our monomer, dimer, polymer naming scheme. A single monomer of a protein is called an amino acid. This right down here is one amino acid. An amino acid has the same structure, and I don't need you to memorize the structure, but you should be able to recognize it. It has a central carbon. They always have this NH2 group, that's an amino functional group. That's where the amino part comes from. And then it's got this carboxylic acid group over here, which is where it gets the name acid. You can see down here it's got like this R. So R is the variable group. There are 20 different amino acids, and all of those are over here. You do not need to memorize the names of these. But what you can see is in this little region that's boxed off, that's the R group. So for each of the 20 different amino acids, the R group is different. And when you have a different structure, that gives you a different function. So the ones in this yellow box tend to be nonpolar, meaning that they don't mix well with water. The ones in this blue box tend to be polar, which means that they do mix well with water. These purple ones have a negative charge. These green ones have a functional group with a positive charge. So all of that's going to give them different functions. The dimer is called a dipeptide, and a polymer can be called a protein. Sometimes we call it a polypeptide also. Proteins have 4 different levels of structure. The first primary structure is just the amino acids strung together in a sequence, kind of like the letters that make up a word. If you changed the sequence of these amino acids, you would change the function of a protein. Just like if you shifted all the letters in a word around, it would change the meaning of that word. These amino acids then interact with each other to make a secondary level of either alpha helix or beta pleated sheets. These secondary structures, the helixes and the sheets, are folded up into a large tertiary structure. So this is the kind of folding of the overall protein. This gives it its 3D shape. And you can see that this is all one chain, right? I can follow this protein all the way around from one end to the other. So this is one peptide, right? Or one polypeptide. The quaternary structure is when many of these 3D shapes get put together. So if you look down here in this example, you can see 1, 2, 3, 4 different peptide chains, and so that's what gives it its quaternary structure, and it's now going to be functional. So take a second, it's literally a minute, 51 seconds to watch this video. I'll post a link in D2L about showing you kind of how these structure levels actually interact. Enzymes are an example of proteins, right? So we're still talking about proteins. An enzyme is a really specific type of protein that's called a catalyst. And what this does is it makes reactions go faster. And it does this by lowering something called activation energy. So here's a reaction, right? This is the amount of energy we start at. It requires some energy to be put in to start a reaction, and then this reaction is going to go. Think about rolling a ball down a hill, right? Here's my hill. Here's my ball. I'm going to roll a ball down the hill. The ball doesn't just roll down the hill on its own. You have to actually push it. And once you get it over the hill, it'll go. Which hill do you think is going to be easier to roll the ball down? This one's a little bit higher, and this one is shorter. Yeah, this one's going to be easier. This is the one that has the enzyme. It basically makes it easier to start the reaction. So here's my enzyme. In this reaction, I want to break these two apart. So these are going to enter what's called the active site. The active site is going to put stress on this bond here, and then the chemical reaction is going to occur. One thing to keep in mind when we talk about all of our energy or all of our metabolism is that every reaction we're going to see has an enzyme associated with it. Enzymes are very specific. This enzyme is only going to work for this specific substrate. And enzymes are reusable. Once this product is released, we can go right back to step one and start a new process. The last and final group of macromolecules, so fourth group here, are called nucleic acids. A single nucleic acid is called a nucleotide, and a polymer of many nucleotides is called a nucleic acid. It's the same name as the group. So some examples are DNA, RNA, and ATP. These are really, really important. So DNA is your genetic code. RNA are kind of short temporary copies of the genetic code that we use to make proteins. And ATP is our energy molecule. If we were in a classroom, I would be jumping up and down. It's how we make cellular energy. So nucleotides all have three specific parts. They have a sugar. So if we look around here, this is one, it's got a sugar. This one's got a sugar. Here's my sugar. There's my sugar. They have a nitrogenous base. That just means it's got nitrogen in it. Right? This has nitrogen, 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 nitrogen. And they have at least one phosphate group. So this is ATP right here. This has 3 phosphates groups. Here's my phosphate group here, phosphate group here, phosphate group here. So you do need to know the 3 parts of a nucleotide. So ATP stands for adenosine triphosphate. Adenosine is the nitrogenous base, and the important part is the triphosphates. This is our energy transfer molecule. So here's an ATP, and it's triphosphate because it has 1, 2, 3 phosphates. And one thing to remember about these phosphates is they have a negative charge on them. So imagine taking really strong magnets and trying to stick the negative ends together. That's hard to do. It takes a lot of energy. So when we break this bond here and release a phosphate, it actually releases energy. And we end up with something called adenosine diphosphate. 1, 2. ATP is renewable. I can take energy from our food and put it back into this reaction to make more ATP. So this is how your body stores and makes energy. So we take energy from our food, we make it into ATP, and then we release it to do biological work. So take a moment and reflect on these 4 different groups of macromolecules.